So we mapped the genome out. The human genome was mapped in the early 2000s, and we have a lot less genes than we anticipated. We've got like 23,000 genes, and there was an expectation prior to mapping it that we'd answer a lot of our questions around chronic diseases, that we would have like a genetic answer for why these are occurring. In fact, we didn't. There isn't a clear through line from genes to chronic disease. And that really exploded the era of epigenetics because we saw that it was above the gene. So technically, the term epigenetic means epi, above, genetics, the genetic material, so above the gene. So rather than our genes being the cause of accelerated aging or longevity or our risk for cardiovascular disease or cancer, et cetera, it's not. It's what is above the gene. And that is the interface of environment. So that is how we live our life. So how we live our lives, and we can talk about there's a heritability component in epigenetics as well, we can talk about in a minute, but how we live our lives is filtered here, influences gene expression at the level of epigenetics. There are a number of different biochemical processes that happen that regulate gene expression. The best studied and really arguably one of the most important is DNA methylation. One of the reasons it's the best studied is because we have really good tools to measure DNA methylation, whereas acetylation and changes to the histones, the proteins that the DNA is wrapped around, like we don't have quite the same, or looking at small RNA and their role in gene expression, we don't have quite the same level of reliable tools to investigate the other epigenetic processes. But DNA methylation is special for a few reasons. We can preserve DNA methylation patterns, cell division after cell division after cell division. There's enzymes in the body, DNA methyltransferases, that keep those patterns intact, which is not true with some of the other epigenetic marks. DNA methylation and demethylation, they determine the fate of pluripotent stem cells. So in embryogenesis, methylation is largely wiped clean, not completely, and that's the heritability component we can talk about, but mostly wiped clean. And then that embryo's development occurs via methylation pattern. Is this pluripotent stem cell going to, turn into an optic neuron, or is it going to be a skin cell, or is it going to be a heart cell? It's extraordinary how powerful it is, and it's consistent. So those marks are preserved. Like, you want your skin cell to remain a skin cell, right? You don't want it to turn into a liver cell or something. So those marks are resilient and preserved over time. One X chromosome in those of us with two X chromosomes is methylated to shut it off. And that is clearly preserved over time, over, cell division after cell division. So methylation and demethylation play very fundamental, extremely important and long-lasting roles in the body. It's also very responsive. So other regions of the methylome, as it's referred to, are responding to our daily inputs. And there's a continuum. So depending on, the quality of our inputs, it will dictate, gene expression's fate, for the better or for the worse. So that is a little bit of background. And let me see if you've got any questions or if I've answered yours.
Yeah, that's great. I just wanted to take that further now. And if you don't mind just going into, there's a lot of information out there, even in this kind of arena. And what are some of the big kind of myths or mistakes that you see people making when, using this information from a lifestyle perspective or a diet perspective or a supplement perspective? What are the big kind of myths that you found when working through this info?
When SNP testing became widely available, clinicians were ordering it, and then, consumers can get, a massive amount of genetic data. And that data often comes with interpretation. And, if you've got this SNP, you've got increased risk for this, this, and this, and therefore you should take all of these supplements. I think that there's just gross overinterpretation out there. And people get really anxious about their SNPs. I think the promise of epigenetics is that your genes don't dictate your fate, or very limitedly so. All of us have had patients come to our office with a really high degree of anxiety because they've done SNP testing and they think that they need to be taking supplements X, Y, and Z forever, or that they're going to get diseases X, Y, and Z. I talk about some of that in my book. I think that this was originally kind of speaking to that, like, let's maybe course correct the conversation. So that's one piece. But the other piece is in this era of being able to see gene expression. We're starting to be able to see gene expression. Believe me, in another 5 to 10 years, we're going to have we're going to have more tools. We can see how our diet and lifestyle, our nutrient inputs, our stress level. I mean, we can see how this influences genetic expression. People probably have heard of the agouti mice studies. Randy Girdle and Waterland, in the early 2000s, published the first extraordinary nutrition epigenetic study where they looked at an agouti mice, or a group of agouti mice, pregnant agouti dams, and they're obese and blonde, and they're vulnerable to diabetes and cardiovascular disease because they have this agouti this agouti gene on. So they're very visually distinctive. They look like these little orange blobs. And Girdle and Waterland gave them methyl donors. They gave them B12 and folate and betaine. And lo and behold, they were able to methylate and turn that gene off and really restore the mice to like a wild type. They call them pseudo agouti. They're, skinny little brown, house mice after that. So you can see the power of these nutrients influencing genetic expression ridiculously potent in this study. And I want to point out that those changes in that first generation lasted multiple generations. So it wasn't just the immediate offspring. Those vitamins had impactful results on genetic expression generations out. That's a big deal. I mean, if that doesn't cause you to think about how you're dosing your vitamins. We're not just peeing them out, they're not harmless. Like I was taught that they're water soluble. They're not a big deal. You're not going to store them. You're going to get rid of anything you don't need. Yeah, maybe. And they're also influencing gene expression. They're potent. Waterland and Girdle summed up their abstract in that first paper, and actually subsequent papers they would talk about this, cautioning our understanding of nutrition supplements, basically suggesting that we recognize the power of them to influence genetic expression, and that we be mindful of that. I think that our focus and obsession on single nucleotide polymorphisms and prescribing shopping lists of supplements to sort of remedy the failing of our genes was just really kind of misguided in our approach. And so, again, we created this very, very food forward approach, recognizing the potential to influence gene expression. I wasn't sure that we would reverse biological aging in our study. Obviously, I hoped that we would. It was one of the principal things that we wanted to look at. But I felt certain, maybe it's naivete, but I did feel certain that we would influence genetic expression. I felt certain that we would see some changing to the epigenome, and we have, and we'll continue to unpack and publish on our findings. But the first big finding was that we looked at the genes involved, or the methylation patterns involved in biological aging, and we took them to a younger pattern, which is pretty extraordinary. I think it's cool for a lot of reasons, but it certainly highlights the power of diet and lifestyle.